Okay. All right, so we are to the introduction. So I'm really excited to be having this meeting. Can everyone hear me?
Yep.
Okay, great. So I'm very excited to be having this virtual user meeting. It's our first ever user meeting for the Crystallization Center. We are located in Buffalo, New York, and we are the National High Throughput Crystallization Center. I'm going to just getting my slides separate situated. We're going to get stuff started. I hope. So just for some housekeeping, the meeting will be recorded, and we're hoping to post things on our HWI YouTube channel for people to take a look at later. And of course, if you're speaking in the session and you don't want to be recorded, please just let us know so that we can edit that out or stop the recording. So please, of course, mute your microphone unless you're talking, and you will be muted when you come in, so that should be pretty good. If you have questions for speakers as we're going through the meeting, please type them into the chat, and that way we can ask those questions as we're going along. And we will also have a panel discussion. We'll be talking about that as well. Give me a second here. Okay, so our schedule. This was our original schedule until late last night. And so we had a slight shift because Karen is unfortunate with COVID and is unable to present. And so we've modified a little bit of our timing. And
Sarah,
so
Sarah, you broke up a little bit there. You might want to just repeat what you just said, and maybe check your internet connection or turn your video off for a second.
all right, I'll turn my video off.
I don't know if it broke up for other people,
So, thank you very much, Miranda. So we do have a slight change in schedule. Unfortunately, Karen Allen from Boston University is sick with COVID. And so we've shifted things a little bit. I'll talk a little bit longer this morning, or give a little bit more time for some Q&A. We're going to have a slightly longer break. And that's really pretty much the only change. We will be having a panel discussion at the end of all of the sessions and a general Q&A. And so I encourage you to send questions or discussion topics to us, either in the chat and just say panel, this is something I'd like to discuss, or you can email us at htslab@hwi.buffalo.edu before the panel discussion to suggest topics or ask your questions. Of course, you can do this during the panel session as well, and we can hopefully have a very nice, engaged conversation about crystallization methods and topics and pipelines. So with that, we're going to start right in. And I want to introduce everybody on the Crystallization Center team, on the HTX Center team. And so I will, in fact, start. My name is Sarah Bowman, and I have been at the HTX Center since 2017. And my role in the center is to direct the center. So I've kind of got the job of writing grants and making sure that we have funding to make sure to support the resource and provide the resources to our users, as well as to kind of do outreach and engagement with the community, because we really are liking being a huge part of the structural biology community. So I will turn it over to Elizabeth.
Hi, my name is Elizabeth Snell, and I've been working in the HTX Center since last February. I've been at HWI, however, for a total of 14 years now. I work on pipeline development for optimization, and I'm also working on growing very small crystals, the opposite of what people usually want, but very small crystals.
Great. Let's see, Miranda.
Hi, I'm Miranda Lynch, and I joined the Crystallization Center and HWI in 2018. And I'm a computational scientist and an imaging scientist, so I get the fun of working with all of our imaging data. I work in developing ways of maximizing the way we can use the information content from our imaging. You'll be hearing a little bit about the specialized imaging and stuff very shortly. I'm working with Elizabeth on some of the things pertaining to the microcrystal project, and so we've got a lot of exciting things going on there. So I'm a computational scientist who participates in that aspect of our center.
Great. Tiffany?
Hi, my name is Tiffany Wright. I joined the HTX Center in November 2022. I manage the day-to-day operations in the High Throughput Center. This includes plate setup and the plate imaging.
Great. Gabby?
Hi, everyone. I'm Gabby Bujaszewski. I'm the operations manager for the HTX Center, and I joined the team back in September 2022 after finishing my doctoral work at UNC Chapel Hill. And what I'm primarily responsible for here is working to translate our optimization pipelines in a fee-for-service model that we will soon make available to you all, as well as managing academic and proprietary projects.
Great. And I do want to also just call out a couple of other people who are also part of the Crystallization Center. Steve Potter is our real RT specialist who helps us to kind of transfer images as we're doing those things. And you will hear from a former student of the Crystallization Center, Ethan Holloman, this afternoon, who's now a doctoral student at UC Davis. But he has actually been the person who has developed the Marco Polo GUI that he'll be telling you a little bit about this afternoon. Okay, so with that, I am going to kind of dive right into just introducing the Crystallization Center. And if you have questions, please do type them into the chat. And so just a little bit of background. So in terms of history, we have been a high throughput crystallization screening center, and we've been running for over 20 years at this point. The Crystallization Center was established during the Protein Structure Initiative. And so we were the kind of main source for crystallization for a number of the different crystallization, or a number of the different PSI initiative sites, including the Northeast Structural Genomics Consortium, or NESG. We'll be talking about a couple of these. We have a really pretty good rate of crystallization success. As many of you know, crystallization is one of the major bottlenecks in structural biology and crystal-based structural biology. And so what we've done is really create this high throughput setup as part of the PSI that we're now offering to users from all over the world, giving you a sense of kind of how this has kind of worked. For NES 170 distinct constructs of a little over 2,800 proteins. Of those, close to 50% we were able to get crystal hits, and of those, actually, that number is a little bit higher at this point because structures are still being solved. A few over 700 protein structures were deposited to the PDB. And so this means that this is a really efficient way of actually finding crystal conditions. Okay, so
Go ahead and turn your video off, Sarah. You are still having some audio issues while your video is running. So there you go.
okay, and we can go ahead and have everyone turn their video off if you guys would like, so that there's just a little bit more bandwidth internet. So who uses the Crystallization Center? So we have users from academic, nonprofit, government institutes, as well as from industry. We use a fee-for-service operations model, and in 2021, NIH gave us funding to become a national resource, which provides subsidized support for non-proprietary samples. And so Having troubles. Okay. And so just as a reminder, for anyone who is making use of the Crystallization Center, we do ask that you go ahead and make sure to cite the Crystallization Center grant when you're publishing papers that have benefited, similar to citing a synchrotron beamline or a cryo-EM center and their funding. We'll do this again as we're kind of moving on. I'll keep reminding you of this. So who are our users? You know, maybe you want to efficiently and rapidly screen through 1,536 non-redundant conditions. So that's similar to 1696 well trays. If you want responsiveness, security, and confidentiality, if you want to be able to distinguish protein crystals from salt crystals, we're going to talk about that a lot this afternoon. You want to detect submicron crystals. You want to auto score your images of crystal growth. Again, we're going to talk about that a lot. Who else are our users? Maybe you've already tried all of the screens in your lab. Maybe you manually set up all your trays and you'd really like some access to robotics to help you do that. You manually inspect all of your wells for crystal growth using a microscope. You don't have advanced robotics or imaging, and you really need fast, easy access to screening a wide range of crystallization parameter space. Okay, so since we actually received the funding for the Crystallization Center to become a national resource, we have set up close to 300 samples for over 90 different academic, government, and nonprofit research groups, and that is located at 70 different institutions. So this doesn't include any of the proprietary samples we've worked with. And at this moment, for the past year and a half or so, we've worked with people from 27 states, the District of Columbia, and 2 other countries. So we really want to be a resource for the structural biology and the biology community to be able to provide access to structural biology. Okay, one of the things we're really excited about with regards to this meeting is engaging with all of you to find out what are your crystallization bottlenecks, what are the things that you would be interested in having access to, and what services would you like to be available at our centralized crystallization center. And so please feel free to add questions, add comments, just ask what the things that you're interested in. And I'd like this to really be a conversation with our users, potential users, other crystallization people who are working in this field, because it's an exciting field to be in. We will be doing a number of polls and a survey during the meeting. And so I'm going to actually try to start a, actually, I'm not going to start a poll yet. Okay, so I would actually like to start a poll right now, just to begin, kind of get people stretching this morning. And this is, you know, what is the method that you use most of the time for your crystallization experiments? And so I'd like to go ahead and have people answer, and I will of course show the answers once we kind of have some of these. It's multiple choice. Thank you. I see answers coming in. And we'll give it just another 30 seconds or so for people to answer. Okay, here we go. I'm going to share the results of this poll. And so thank you, everybody. This is actually really helpful for us to have, so that kind of have an ability to kind of talk about what we do. Now, it looks like most people are using vapor diffusion and microbatch under oil. Or sorry, vapor diffusion and then some microbatch. Let's go team microbatch, Gabby. I like that. And so we in the Crystallization Center, we use microbatch under oil as our major technique. And so We have this high throughput pipeline. As I mentioned already, we have 1,536 different conditions in one experimental microassay plate. We use the microbatch under oil, and this is our experimental plate that you can see up here. And I think my pointer is working, so let me do that real quick so you can see our plate here. So again, this is equivalent to 1696 well trays in the same physical footprint as a normal SBS format tray. So what this allows us to do is use really minimal sample volume and minimal crystallization cocktail, and we actually put the oil on top and then have cocktail and then protein. We will be kind of at the bottom of the well. We'll be talking about that in more detail shortly. So we provide the robotics, the imaging instruments, and the expertise to really help people. We first of all do bright field imaging, and one of the things we do is we do an image before we add the protein or the sample, and that is as a negative control, because if there is anything in the well that is already crystallized, you don't want to pursue those as one of your options for crystallization. And then we monitor actually over the course of 6 weeks. And our high throughput screening results in a minimum of close to 14,000 images per experimental sample that's set up. And so we also have a graphical user interface called Marco Polo that helps us to sort through that very efficiently. Okay, so this kind of gives you a sense of what we actually do. And this is from a recent paper that we just published in ACTA Crystal D. We make our 1,536 crystallization conditions. We do this every 6 to 12 months, and we store these in 96 well deep well blocks. We then also prepare 384 plates. We store these for 4 to 6 months. We'll go into some of these details of everything, but we essentially fill the 1536 microassay plates with oil. Then we add cocktails to those 1536 microassay plates, image those plates with the cocktail only, and then proceed to kind of adding sample and monitoring those crystallization experiments over time. We will be talking about more of these details as the day goes on. I do want to just highlight that we did get the cover of ACTA Crystal Section D in this month, so it was really exciting to highlight our robotics, imaging, and computing that we're doing here in the HTX Center. So this is another image from the second paper that we've recently published, where Gabby is the first author, and it's in the Journal of Visualized Experiments. And I think it's helpful for people to kind of get an idea of how our cocktails are actually stamped out, because it's quite a bit different than going from a 96 well deep well block into a 96 well plate. We actually stamp out 4 96 well deep well blocks into one 384 plate. We do 4 of those, and then those 4 are stamped out into the 1536 well plate. And so keeping track of exactly where each of the conditions are and mapping those onto the plate so that you know what your conditions are that have generated crystals is one of the key things that that we also kind of really keep track of. And we're going to talk a little bit about that as well. So I will say that this paper is just accepted. The text is available, and we will be filming in April, and that will provide then a real way, I hope, for us to be able to show our users exactly what we do for setting up samples. Okay. The 1536 microassay plate, these wells are actually really quite small. So just to give you a sense of how large they are, this is kind of in someone's hand. We actually image these upside down in the rock imager on a specialized adapter, and that is because that is the best way we can get our images to work well. And so this is just an image of us putting a plate into the imager, into one of the rock imagers to be able to image each of the wells. We do image each well every week to monitor for crystal growth. So So talking a little bit about the robotics in the Crystallization Center. So we have a PlateMate. We actually have a couple of PlateMates. And these instruments have really been kind of some of our workhorses for a long time. They are actually about 16 years old at this point. And so we've been working over the past couple of years to replace those robotics with new robotics that help us to perform what we need to. So we have an Integra Via Flow. We use this, well, we'll talk about everything we use it for. The Formulatrix Formulator, an Art Robbins Griffin, SPT LabTech Mosquito, and also a Formulatrix NT8, which we're just showing you a little video of. Okay. We also have imagers. So we have two Formulatrix rock imagers. We have a Formulatrix rock imager 1000 with Sonic. We will be talking about what exactly Sonic is, but it's essentially a way to help us to detect crystals very efficiently. We also have a rock imager 54 in the 14-degree room. We do 4-degree imaging using this imager as well. We have a Mitogen watershed for humidity controlled harvesting of crystals, and this is an amazing instrument. And of course, we have pucks and shipping doers to be able to send crystals to synchrotrons. So I want to actually highlight the things that we are kind of new to the Crystallization Center, because I've been here for 5 years. We've been doing a number of things where we've been changing kind of some of our operations. So one of the new things is that we prepare all of our in-house cocktails using the Formulatrix formulator. Our newest generation of cocktails have actually been formatted onto the plates in a more efficient way. And so we will be actually putting out the new generation 21 of our cocktails as of April of this year. And so our cocktails, we like to say that it's that our cocktails have turned 21, and and this is our little cocktail maker. One thing to be aware of for those of you who have used the Crystallization Center in the past is that this means that the well positions that cocktails have been in have moved around a bit, but that will all be mapped onto the different software that we use and we provide for people. Okay, so we actually add oil with the Integra Via Flow 384, and we actually stamp out from the 96 well tray into the 384 tray, also with the Integra. We add cocktails with Art Robbins Griffin into the 1536 plate. And newish, a couple of years old now, is that we have a sample submission form online. Okay, as of just this month, we have started doing sample addition with the SPT LabTech Mosquito, which is shown here in the lab. So this is faster. So first of all, it takes only 4.5 minutes to dispense the sample to the entire 1536 plate, and that's in a humidity controlled environment. And so that's actually really great. It's about it takes about half the time that it used to take using the PlateMate. And even more importantly, it's really efficient. It requires now only 350 microliters of sample, and this is down from 500 microliters of sample. And so we're really excited that we can actually be doing all of this high throughput setup with even less sample than we've had to use before. And we're going to be doing some tests to kind of explore that a little bit more to see if we can get even lower. Okay. Some of the things that we like to do in the Crystallization Center is really feature success stories for center users. So one of the things I'd like to tell all of you is that if you use the Crystallization Center and you have a success story that you would like to share, please let us know, because we like to feature that in our Crystals of the Month. We don't end up having features every single month, but we'd sure like to. So I'm just showing a couple of examples. In this case, Megan Shelby actually worked on growing crystals of 2 of her proteins for nanolipoprotein particles for some initial XFEL-based structural studies. And so it's always really nice to see things in the high throughput screen then scaling up. We like to put out messages like Happy New Year in 2022. And of course, we actually did a lot of work during the pandemic and highlighted a number of different things here. This is from Andrei Kovalevsky and Daniel Neller, some of the first structures of the SARS-CoV-2 main protease, which we crystallized in March of 2020. So it seems like a long time ago now, but we also have featured recently Marina Prima and the Izzard lab at Scripps with this was the only condition that they were able to get crystals to form, and they were able to optimize those conditions in their lab and move forward and solve a structure. And then finally, we have just December 2022, a number of different conditions were found for this pseudo oxynicotine amine oxidase from the Bardwell and Stolt groups. And that structure is just out. You can see that many of these crystals are actually underneath precipitate, but you can see them in part because we have UV TPF. And again, we'll be talking about the specifics of some of the imaging resources that we have a little bit later. Okay, so one of the things that I would like to really just point out is if you have something new out there that was screened in the HTX Center, please let us know, because we love to feature new results. Okay, how do you access the high throughput center? Well, for the high throughput 1536 screen, we actually run things on a schedule. So we have, I've listed the website here, and I think it's going to show up in the chat as well. On the website, we have lists for when our screens are happening. So you can see here that we're currently taking reservations for the soluble and the membrane screen for April, and those will be running in April. We typically have those on dates and figured out what dates are going to work. We also have an announcement email list. And so if you're interested in signing up for this announcement email list, you can actually send an email to this email address. And again, I think that we will pop that into the chat, and then respond to that confirmation email that you get to get your email added to that list. Okay. So we really appreciate everyone attending and participating in the user meeting. Want to remind people to cite the grant in the acknowledgement section of publications, and also cite one of the papers. If you're using the Crystallization Center, it helps us to kind of maintain our funding to make sure that we are able to kind of show results from the work that we do. We will be having a user survey that will come up, and we'll link that in the chat. It will also be emailed out. I want to also highlight that we will be putting in an NAHS 10 grant for a new sonic instrument, a new rock imager with sonic. And so if you are interested in helping us with that, please let us know. We are looking for users who might be interested in helping us work on some of that. Again, I want to remind you that we have panel discussion and a general Q&A. So if you can please send questions and discussion topics to us either in the chat or email us. I want to just remind you what our schedule is. And for those of you who signed in late, we did have a last minute cancellation because of COVID. And so we've shifted the schedule around for a little bit. I do want to highlight again, all of the people who kind of really make things happen, our resource funding, as well as the resource funding that we were able to make use of during the pandemic for SARS-CoV-2 related samples. We do have some research funding as well that helps us to do some of our microcrystallization work, as well as work on antibacterial and metalloproteins. All the people who are kind of involved, and pictures. And because we have a little bit of difference, a little bit of time, I'm also going to move forward and just tell you a little bit about some of the other work that we've been doing. And don't worry, I will leave some time, a couple minutes for questions. I want to highlight, I'm hiring a postdoc to work on microcrystallization technology development. And so if you or someone you know is interested in doing these types of things, please do sign up, or let me know, reach out. And again, just because we had a last minute cancellation, I thought I would talk a little bit about some of the kind of research work that we do. And so I'm going to highlight some of the work that we've done with another student that is not at this meeting, and that is, what does it mean to grow good crystals? So we often think about growing good crystals as getting bigger crystals that diffract better, often through some type of optimization, and that's stuff that we are actively working on in the Crystallization Center. It can also mean figuring out good cryo conditions to get crystals that diffract better, again, often through optimization. We think that there's other things that we can also be doing. We could better detect crystals. And so that we think that there are some machine learning algorithms and additional imaging methods. There are using, and I'm going to actually focus first on using the data that are already available and looking at the crystallization details that are available in metadata on the PDB to help us design new screens and mine data to understand patterns. And finally, given a protein sequence, can we predict conditions that will actually yield crystals? And so I think that is something that one of our speakers later in the afternoon is going to actually speak about. So I'm excited. So I want to just tell you a little bit about a project that we were working on, where we were looking at crystallization metadata in the PDB. As those of you who've submitted samples know, there's a free text field in the PDB for crystal growth condition metadata. It was previously called Remark 280. It's now experimental crystal grow.pdbx_details. And it's a rich data source for mining successful conditions in crystallization, except that it's a little bit inconsistent. So I had a student, Max Dudak, worked with me a couple of years ago, and he made a tool to actually parse that free text field into sensible structures. And we've created a database. And so what you can see is that we can go from what's in that field, we can parse that into kind of meaningful information, and then actually have that information available. And we've, again, created this database that is available for people to make use of. And you can see that sometimes there's even more information that's provided, and more information is really great as long as it can be machine parsable, which in this case it was. So we were able to pull from this Remark 280 field all of these different conditions that were important in the crystallization cocktail for this particular sample. Unfortunately, there's also many nonsensical details in the metadata in the PDB. And so you can see that in this case, in the top case, we see that it was that whatever the conditions were, it was vapor diffusion, sitting drop, and the temperature was 293. So in terms of trying to kind of reproduce something, that's actually not meaningful and not helpful. And it's also not really helpful in terms of doing any data mining. And in this case, these conditions were screened in 1,200 different conditions and other crystallization conditions identified using literature and data. And so again, no details are provided. And so the dictionary that we made does not recognize any of these words as chemical components. And so there's a variety of different ways that things can kind of go wrong in terms of how you're entering your PDB information. We did get the cover of patterns when we put this paper out as well. So one of the questions that sometimes comes up is it possible to add crystallization metadata to files that have already been deposited? And so I want to tell you all right now, yes, you can certainly do so. You can version your PDB entry to actually add those details. And it's really important for people who are doing data mining and data entry, using that metadata to actually have that available. And so I hope giving you a little bit of a taste of some of the things that we're doing with regards to kind of using data that are available already. And, with that, I would like to kind of wrap up my talk and open the floor for questions or discussion. And so I'm going to attempt to stop sharing. Okay. All right. So Are there any questions, any comments, anything that you guys are interested in talking about at this point? I know that it looked like we did have a question about what type of oil. Doug, as Gabby mentioned, we use paraffin oil, and it's a very high viscosity oil. And that in part is because we need the oil to stay in place, first of all, over the course of 6 weeks, to make sure that the drops don't evaporate. And secondly, we need to make sure that the plates themselves can be turned upside down without anything dropping out, because that's how we image them. And so if we have the specific type of oil that we can give you the part number for that if ever you would like to use them. And we do optimize with microbatch with that oil as well. So any other questions? If not, I'm going to put another poll out. Okay, keep in. Great. So is there a service to predict crystal conditions according to protein sequence? There are some services, or there are some software available to be able to do that. I can say that maybe not conditions, but maybe crystallization propensity. And I believe that we're going to get a sneak preview of some new software that's coming out to do just that this afternoon. And so I encourage you to stay and hear about that. Let's see. I'm going to set up another poll, and I'm sorry I didn't. And when you're setting up crystallization experiments, what methods do you usually use? And we're interested in kind of finding out about what people are doing.
Sarah, there's another question in the chat from Anwar. They ask, is there any way to minimize salt crystals?
That is a great question. So there are some things you can do, right? So first of all, one of the recommendations that we make is to make your buffer conditions to be as kind of for your protein sample to be as low concentration as your protein can handle it, in part because a lot of the buffers that we use, like phosphate buffers and things like that, actually will crystallize very easily. And so that is one of the things that we like to do. For us, we also pay a lot of attention to whether we're getting protein crystals to form or not. And again, there'll be a little bit more about that this afternoon. We use things like UV TPF or UV Vis to be able to tell if the crystals that we're seeing are in fact protein crystals or salt crystals. Gabby, you want to join in and add any answers to that?
No, I think you mostly covered it. There is another question now in the chat from Doug. He asks, you were talking about microcrystals. Any plans to link microelectron diffraction to your microbatch hits?
Yes. And actually, we're interested in we're really trying to build out some pipelines for finding these microcrystals and then using them for microcrystal electron diffraction. In part, because we're able to with some of our imaging actually see some of the submicron sized crystals. It's really exciting. It's an exciting time to kind of be doing this and to have those capabilities, because we have some possibility of being able to identify these crystals and then move them into a pipeline for microcrystal electron diffraction. We're not quite there yet. It's part of our R01 research funding to be developing these pipelines, but we are working with the Cryo-EM Center, which is conveniently located one floor down from us. And so Devram Hehan and Katie Spoth have been working with us to try to develop some of these pipelines. It's in, I would say, development at the moment. I'm going to end this poll and share the results. And so it does look like many people do use robotic drop setters. And one of the things that I think is important to kind of keep in mind is that a lot of the robotic drop setters and the methods and techniques that we use at this point out of hand that are just normal for a lot of crystallization labs were really developed in the PSI kind of time period. And so I think that there's a lot of manual, a lot of kind of resources that have been developed that are now available because of the PSI. And I think we'll go ahead and Fernando, nice to see you. I see your question here. What is the most common reason that users may have trouble scaling up crystallization conditions manually in their own lab? That's such a great question. So when we do scaling up, we will often try vapor diffusion. Vapor diffusion isn't always going to be successful when we've identified crystal hits in microbatch under oil. And so we advise doing both vapor diffusion and microbatch, maybe not at the same time, depending on your sample, how much sample you have, but also, but at least sequentially. If it doesn't work in vapor diffusion, try it in microbatch. It can also be hard if you robotics to be kind of precise about kind of being able to generate a grid screen around the conditions that were initially successful. There are a lot of things that can make a difference in crystallization, as many of you size of the drops, to the temperature, to the humidity, to the exact chemical composition of your cocktail. This is part of the reason why we're really interested in and why we're pursuing a lot of ways to start being able to do optimization and scale up for our users, because then essentially you would have access to the exact same cocktail components that you started with in the high throughput screen, and then we can set stuff up there. You know, Gabby will be telling you a little bit about some of those optimization protocols and pipelines. We're in the process of introducing those. Okay, so I think that with that, I want, please feel free to keep asking questions and ping us with questions. I'd like to, at this time, actually, Eric, I think I saw you here. I'd like to introduce our next speaker. Hi, Eric. So I actually got to see Eric just a couple of days ago. He gave a great talk at ASB&B. I'm really excited that he agreed to come and give this talk today. He's been a user of the Crystallization Center for some time, and he does a lot of really cool structural biology work. And so thank you very much, Eric. Do you want to go ahead and share?
Sure. Thanks,
Great.
Sarah. I appreciate it. So I'll just go ahead and share my screen here. We all seeing this? In the middle, which is not where I want to be. Okay, we good?
We're good. It looks great. Thank you.
Okay, great. Well, thanks, Sarah, and thanks everybody for being here. Also, have to thank the Hauptmann Woodward Institute in general. It would have been really difficult, I think, for a relatively small lab at kind of a small school to really be able to do some of the structural biology stuff that we've tried to do without it. So I solve structures with HTX, and so can you. That's my cheesy title there. Anyway, those are some of our hits there that we've had. So I'm just going to kind of go through some of the projects we've done, just in no detail, just to kind of highlight how HWI has helped us out. So I started getting into crystallography as a postdoc, working with Dr. Carrie Wilmot there at the University of Minnesota. And I walked into a project where this super cool structure, which I apologize for the resolution there, had already been solved by a previous postdoc, Lyndall Jensen, of an enzyme, the methylamine dehydrogenase in blue and green there, and then the heme enzyme that's responsible for the maturation of its cofactor. So here, I don't know if you can see my mouse there, but you have this these two tryptophans, which are then oxidized to form a quinone, and that requires this heme. So this is in sort of a premature state. So since there was a structure already, I didn't have to try to search for conditions. And basically I was looking at intermediates and some mutations and things. So if we kind of zoom in on the electron transfer pathway here, you can see that you have, do I have a little laser pointer? There we go, cool. Here's our site that gets oxidized to form the TTQ, tryptophan, tryptophylquinone, and it's interspersed here with tryptophan residue on the heme enzyme. There's our low spin heme, another tryptophan, high spin heme. So this thing is getting oxidized to what we call a bis iron 4. It's a really potent 2-electron oxidant, and then it's just pulling electrons out one at a time from the site. So one of the things that I did was to try to see intermediates in that process. You have 3 2-electron oxidations occurring, and basically all we did is let those crystals sit in their crystallization drops and oxidize over time. Turns out PEGs will form peroxides, especially in light, slowly. And so we could just kind of let them hang out. And then as you can see, over 20, 30, 40 days, you get this cross-link formed. And then later on, you start seeing this oxygen atom appear there to give the mature TTQ. So this really got me into crystallography. It was a cool project. It was a lot of fun. There were big, red, beautiful crystals, which is always cool. And then when I started my, actually even before I started my independent career, we had this sort of what I call a happy accident here. At the time, I wasn't super pleased, but what we got was this colorless MAUG mutant. So we had a collaborator that produced these proteins from the host organism, Paracoccus dinotrificans. Those are C-type hemes. They need all the machinery to put them in. You can't do it in E. coli. And he sent us this mutant, which looked fine on an SDS page gel. It's about 40 odd kilodaltons, which we expect, but had no color. So we were like, wow, that's amazing. How did we, produce a mutant that doesn't have hemes? Well, I didn't know because I couldn't crystallize it because it wouldn't crystallize under the same conditions. I had no other metals. And finally, I did the mass spec out of frustration just to see what this thing was. And as it turns out, it wasn't MAUG at all. It was a paraplasmic protein of unknown function at that time, which was just gene PDEN 1598. It was found adjacent to transporters of zinc. So these ABC transporters, which I've shown in cartoon here, that have an ATPase, a permease, and a solute binding protein. And it was pretty much always in that context, but there was really no information about it. And so this was the first time that I used HWI. We got these nice hits you can see here, nice single crystals that show up in the UV. They even diffracted pretty well, and then I couldn't do anything with them at the time. And so I just kind of let that sit for a little bit, and we'll come back to it. Because then I got a job in New Mexico. So at Las Cruces, which is not too far from El Paso, Texas. So these are the Organ Mountains shown here. And then we were able to get beam time at ALS and start just trying to crystallize other components of the system. So our first successful one was on the solute binding protein right here. This is AZTC. There's the structure, reasonably good resolution. Again, we used HWI to identify hits. They grew these massive plates, which diffracted pretty good. They suffer a little bit from disorder, as really big crystals sometimes tend to do, but they were pretty decent. And then we were able to get the structure shown here. We crystallized it in the presence of zinc, as well as when zinc had been removed. And then what you can see is this long loop here, which is typical of zinc solute binding proteins, but is almost never observed in the crystal structures because of that mobility. And we were able to resolve it here. You can see the electron density is not amazing, right? Because again, it is a highly mobile area, but you can still see it. And then it's adjacent to this zinc site here. So we never really figured out what this thing is for. You can cut it out and the protein still works fine. However, it no longer interacts with its chaperone, which I'll talk about here a little bit later. Okay, Other structures, so we were able to use this to look at how structure changes upon zinc binding. So the blue structure here is the apoform, and then we see the zinc binding site shown down here. And then the green structure is the holo. In addition to that big flexible loop, which we don't really know too much about, there's another loop we call the Z loop here, which actually closes down over the zinc site when zinc binds. And then you can't really call this a coordinate bond. It's like 3 angstroms maybe, but certainly electrostatic interaction between the carbonyl of a valine and the zinc ion shown there. And another kind of cool thing is it looks like the apoform binds some kind of cation down here, which we modeled as a sodium. It's definitely not a zinc because it has no anomalous diffraction, which you can see really strongly here. Okay. So that AZTD kind of sat around for a couple of years, and we could grow crystals of it and they would diffract, and that was all great, but there were really no good homologs in the protein data bank. Molecular replacement consistently failed. So we tried a few things. So it binds zinc, right? So we tried to use the anomalous signal from zinc. Unfortunately, it only binds 1. It purifies with 1. It will bind up to 2, but it's a 40-some kilodalton protein. You just don't get enough signal. And same with selenomet, right? It has 2 methionine residues. So that didn't help a lot either. We could make the selenomet derivative, but the anomalous signal was still very wimpy. We even made some mutations of isoleucines, switching them to methionines and then making selenomethionines out of those. The expression just started tanking. So it was problematic. And then we ended up going, I guess, kind of old school and just throwing heavy metals at it and trying to solve it by single wavelength anomalous diffraction, which eventually did work for a lead, I think it was lead acetate is what finally bound. And here I'm just showing the anomalous scattering from the lead sites for that, and then the initial density modification map that we were able to make for this protein. And so, yay, we got it to solve. That's at 2.3 angstroms. The native was quite a bit better. We also have it from 2 different organisms. So this CK is Citrobacter cocceri. PD is Paracoccus denitrificans. That's the original one. We actually moved to the Citrobacter, and I think we used HWI there as well to identify conditions, because as you can see, the diffraction was quite a bit better, and it was easier to work with. So anyway, that worked finally. We were able to get a structure. I have a little movie here showing it, so it's really pretty, at least. So it's this beta propeller. There's 2 zincs bound, which we confirm with solution binding studies. And then I think, hopefully we're going to look at the active site here in a minute. These have been hanging up today, so I apologize if that's going to be the case. Yeah, it looks like it is. Huh, well, that's obnoxious. Okay. Well, anyway, sorry about that, but I had a backup. I can show all the salient features here in a static image. Pardon me. So if you look down the sort of the barrel of it, this little cloud here is intended to represent solvent accessible area or volume. We have this site near the mouth of it we call site 1, and then site 2 is on this kind of funny little extension of the barrel or the propeller. Here's electron density at those 2 different sites, right? You got 3 histidines in a water, 3 histidines in aspartate, pretty typical, and then the anomalous diffraction. So a pretty neat protein structure. We also knew that this was able to donate zinc to the solute binding protein AZTC. So we've done a lot of, we used a fair amount of fluorescence and other techniques to monitor that process. And now that we had both structures, we could try to put them together to see how that transfer process works. And so we used this program Haddock and developed this structure shown here, where that long flexible loop of AZTC actually protrudes into the central cavity of AZTD, and that places this zinc 2 site, which is actually, we do know from mutagenesis is where zinc is transferred, right where it needs to be for transfer to AZTC. So it's consistent with all of the solution data, but it is of course just a model. This is probably another failed movie. Oh, maybe. Okay, there we go. Nope. It's only doing a few seconds. That's so weird. Okay, again, I kind of anticipated that. So here again, showing the full structure, quite a lot of interaction interface there. And then this is the zinc transfer site from AZTD. This is where it'll end up on AZTC. So really very close, very similar coordination environments as well. And then some of these loop residue interactions that we know are important again for mutagenesis, we've highlighted here. So there's a couple of histidines and a tyrosine there. And then finally, that interaction relies on that Z loop, as we call it, that closes over the zinc binding site. That has to be out of the way. And so it wouldn't occur between a zinc bound AZTC and the AZTD, which is good because that would be pointless, right? We already have zinc there. Okay, so we also were able to more recently structurally characterize another zinc solute binding protein called ZNUA. This is again from Citrobacter cosseri. This is kind of the more cautionary tale, I guess, of crystallography and crystal screening. So we got these hits and there were loads of them. I mean, there were like 25, 30 hits. And some of them were just spectacular, like this beautiful plate cluster here. And they diffracted very poorly. We went through a lot of different of those conditions, trying to reproduce them in house. They always looked pretty good. And then you put them on the beam and they were pitiful. So we ended up using an additive, this polyoxo tungstate tellurium thing, TEW. You can find it in the literature. It's been used actually a bit for trying to stitch together unit cells, essentially, and create better diffraction. And so we used that. It still wasn't great. I mean, the best we did was 3.15 angstroms. Made for a snazzy looking structure, though. This is a couple of unit cells showing each of those polyoxo tungstate clusters parked there at the interfaces. And then the structure of the protein itself looks a lot like solute binding proteins. They all look pretty similar, in fact. And there's the zinc site, which is in fact different from the AZTC. There's a glutamate here rather than an aspartame. So yeah, what I've found is that beautiful crystals don't always, equate to beautiful data, as I'm sure many of you have also experienced. But nevertheless, we were able to get some information out of it. Okay, now for something completely different. My old friend Victor at UCF started collaborating with my lab on a project using a different quinone-containing enzyme called GOXA, because it's a glycine oxidase. This uses a cysteine tryptophyl quinone, so named because of the cross-link between the cysteine and then this tryptophan again is making a quinone cofactor. The reaction glycine, it goes through a few different intermediates. It produces formaldehyde and eventually ammonia, and then regenerating the quinone here. So there's a factor to aminoquinol, and then you got to pull the electrons out of it to get back to the quinone. It actually uses oxygen to do that. So Victor is an excellent protein biochemist and enzymologist. He had a bunch of kinetic data. He sent, as is typical of him, extremely high quality sample. We just sent it then to HWI and got these really beautiful crystals, great big ones. And they diffracted very well, in fact, but again, molecular replacement fails because this is sort of an unusual protein that did not have good homologs. We went back to our old playbook and we used lead again to do this single wavelength anomalous diffraction to yield the structures here. This one is a tank, so it's about 90 kilodaltons as a monomer. It forms a homotetramer. So building this took some time. You can see the 4 different chains all colored here differently. It's really kind of more like a dimer of dimers, however, where you've got this really kind of cool, it's not exactly a domain swap, but you have a ton of interface in between, and a loop from one monomer reaches in to the active site of the other. So this is the CTQ cofactor, pardon me, which you can see diffraction is really nice. And then very nearby are two residues on that loop from the neighboring protomer. So this is highly cooperative, as you might imagine, from all that interaction between monomers. And again, pretty decent diffraction. So we're at about 2 angstroms. The coolest part of this project, I thought, is that so these crystals are bright yellow, shown here. That's because of this cofactor again. And then if you add glycine to the crystallization medium or to your cryoprotectant before
Or
you,
so I should just call.
sorry. So if you add glycine to this, they turn blue, right? So obviously something's happening at the cofactor. If you look at the active site of this crystal versus that one, here's our CTQ, a couple waters are nearby, and here it is, a covalent adduct formed with the glycine substrate. So that was fun. We then proceeded to do just a slew of mutants. Only a couple are shown here. And I kind of just want to highlight what we got out of them. So there are a few really important amino acids in the active site, of course. This aspartate 678 turned out to be very important. You need an active site base at a couple of different steps. First of all, down here to activate this glycine, you need to deprotonate it so that it can come in and form this what we call a substrate shift base. This will need to be deprotonated again, right, to form the product. And then finally, a water molecule needs to be activated for attack at this carbon. And so we think the aspartate does the lion's share of that. If you mutate that to alanine, it completely kills the activity. What's cool though is if you soak those crystals in glycine, it still shows up. It gets to the active site, but it's all kind of dressed up with nowhere to go here. It's really nicely stabilized by hydrogen bonds between those two neighboring protomers, but it cannot react with the CTQ. And so what we think happens, and this is kind of a Franken structure between a few different things, is that this aspartate has to be here through a water to deprotonate this for attack. We form the shift base. It's actually the substrate shift base, which we know because the geometry would be different. Also, we've done some resonance Raman with another collaborator. And so we've also seen that this aspartate can kind of swing in and out, right? So it swings in, deprotonates, forms a shift base, switch out, swings out to then kick that proton out to a big old chain of waters that goes to bulk solvent. The next step of the reaction can occur where it's going to activate another water molecule here where it's going to attack. And we were fortunate enough to crystallize another mutant that stabilized that, what they're calling a carbanolamine or aminoquinol, you could say, or hydroxyamino, anyway, where there's an oxygen atom now inserted here. And that was trapped by mutating this neighboring residue, this histidine, which is normally in the wild type, to an alanine. And so you can't quite activate it far enough to get past that hydroxyquinone step. So that was fun. That was a cool project. We were able to solve a few structures for various mutants and catalytic intermediates, which is cool. Okay, the last thing and the newest thing I have is this so-called Zerg A. This is part of another ABC transporter for zinc. It's unusual in that most ABC transporters have these 3 genes, a solute binding protein, an ATPase, and a permease. This one has a couple of extra DUFs, domains of unknown function, and the Zerg A doesn't look anything like, at least sequentially, any of the solute binding proteins that we're familiar with. So it's an unusual system, but appears to have evolved for the same function, to import zinc into cells. So we took that Zerg-A. If you just look at sequence space, this is a sequence similarity network. There are loads of Zerg-As all over the place. Also in various pathogens. The one we're looking at is in Vibrio cholerae. There's another one in Pseudomonas aeruginosa, and then some of these Aeromonas species that are emerging pathogens. And so there's some interest for it there. We expressed and purified the Zerg-A. We did a wild type, which did not do super well. There's a very long sequence of histidines and aspartates, about 60 amino acids actually, that is probably highly flexible and doesn't make for good crystals. So we cut that out and we're able to get some pretty decent hits that we were able to then optimize and solve the structure. Again, we have a zinc-free and a zinc-bound one, so that's always nice. The overall structure is shown here. It's not a dimer in solution, at least. However, in the crystal structure, you can see that a couple of these zinc sites are actually shared between monomers, which is most likely an issue of crystallization. Again, this is the mutant protein. We see 3 zinc binding sites. We call site 1, 2, and 3. That's consistent with our solution data for that. So animal or classic or typical, I'd say, for soluble binding proteins for zinc. We had to actually soak zinc into the crystals in this case rather than co-crystallize. Only the apo would crystallize. And then if we tried to crystallize the holo form, it just didn't have to take crystals and soak them. They don't love that. So we lose some resolution, but you can still see them pretty well. This is just showing the 3 binding sites that we identified. Unfortunately, we were a little slow and got scooped by an Italian group that solved what's called PA4063. Is the Pseudomonas aeruginosa version. Looks virtually identical to our protein, so it's shown in pink, ours is in green. However, what's interesting is that ours binds 3, theirs binds 2, only 1 site is conserved between them. And also the affinities that were measured are startlingly different. So they used ITC to calculate dissociation constants in the micromolar range. And we were seeing sub-nanomolar up to mid-nanomolar for those sites. So I think there's a lot to learn in this family, and some probably really unusual mechanistic features to be found. Speaking of, we solved the apo and holo versions, hoping to see how some structural rearrangements might be recognized by the permease, and we don't see that really. The apo is cyan, the holo is green. They're superimposable virtually. Little tiny motions to the helix here, a couple of side chain rotamer changes, but that's about it. And we also don't even know which site is actually transferred to the permease, and the permease is super weird. The closest homologs to the permease part are these MACBs, which are macrolide exporters actually, and they look wildly different from what we normally see in zinc solute binding or zinc ABC transporters. They have these big paraplasmic domains, only 4 transmembrane helices. So this whole system, I think, is going to be a gold mine of new stuff about zinc transport. Okay, just to highlight that, I'm showing what little is known about the classic sort of canonical zinc ABC transporters, where you have 9 transmembrane helix dimeric permeases, ATPase on the inside, and then solute binding proteins out here. There's only one structure of these. It's actually a manganese transporter. And then the MACBs, where these are mostly alpha fold predictions, but you can see that they're not remotely similar. Similarly, on the same page, right, the solute binding proteins don't look anything like each other. So right now we're interested in getting structures for and maybe functions for these unknown, these domains of unknown function. We're also really interested in this paraplasmic domain of the Zerg-B, because that's most likely, at least rationally, where the solute binding protein would primarily interact and promote transfer. And we'd love to start looking at the entire system of both of these kinds of transporters. So we've gotten into some membrane protein stuff. We're able to express and purify a couple of these. And I think actually in this month or April, we're going to be sending sample for the membrane protein crystallization screening, which we've never used. and I'm pretty excited about. So with that, I'll just thank my group. That's us here in New Mexico, especially Daniel Valencia, who will be looking for a postdoc job. So Sarah, I forwarded him your link to your postdoc ad. And then this is an undergraduate, Isaac, actually, who's done a lot of the crystallization stuff for this project too. Our funding, of course, HWI, the advanced light source for diffraction, for beam time, and I thank all of you for your attention. Thanks for having me.
Thanks so much, Eric. All right, I'm going to ask you a couple of questions from the chat, if that's okay. But to start off, I'd love to ask you one of my own questions, which is about the tellurium tungstenate additive. I was curious, did it change the space group in your crystals, or did it just improve the resolution?
Well, that's a great question. I wish I remembered. I think this was one where it was one of those weirdos where you'll get different space groups in the same drop.
Oh, yeah.
And the difference is something like C2 versus P21 or something. And I think that the TEW clusters did lock them into one or the other. It did help with diffraction quality. But again, yeah, like I said, not dramatically, right? Which is why this one's an ActorCrys F and it has a 3.15 angstrom resolution. But I think that, if you're having issues like that, it's worth throwing in there. It's crazy easy to make it. So you don't even have to buy it. You can just buy the tungstate and tellurium salts and just cook it up and crystallize it.
Oh, awesome. That's great. I'll have to try that in the future. All right, so a question from Sarah. Do you see movement when the glycine binds? And I believe that this is in reference to the GOX-A protein, Sarah.
It is, and I think you answered it right after I typed that question, so you can just skip that. So
you don't see much. I know that it's
okay.
Quite, the scaffold's pretty rigid. The only thing I see moving is that aspartate kind of in and out. And there may be other more remote things. That protein's so big that there's some surface stuff that goes on, but I don't know if it's really related to substrate binding.
All right, so Anwar asks, hello, Eric. Have you tried annealing with the plate-like crystals that diffracted to 3.15 angstroms?
Oh,
because sometimes that helps.
Right, it can, and I may not have. That's always something I kind of forget about. Because it seems like such a terrible idea, right? But you're right, it does sometimes work. But yeah, I think I did.
Okay.
That's a good point.
And then Sarah mentions another question along those lines. Have you tried any humidity controlled experiments for the poorly diffracting crystals?
Yeah, so humidity control here is a challenge. So our typical relative humidity is in the single digits. So when you do crystal soaks, I try to just be fast and then just do it in a little plate with like a soaked paper towel. So I wouldn't call that humidity control. I guess I would just sort of humidity maximization. But that's a good one too that I have seen where you're kind of talking to where you maybe do like a hanging drop and then you start sort of dehydrating it when you have even already mature crystals.
Yeah, and you can also, there are a couple of beam lines you can do. They're room temperature, but you could do humidity control experiments where you kind of ramp the humidity down and it pulls some of the solvent out. When it helps, it can help a lot, but it doesn't always help. So just something to like think about. So
right.
And we do really love our watershed from Mitogen. All right, Doug says your haddock models of the AZTD and C complex were cool. Did you try crystallizing them together? Flexible loop should be buried.
Yes, we did try that. That never worked. The complex is likely transient based on what we've seen from solution studies. So it's kind of a cool system because AZTC has a single tryptophan that's close to it's like down here, it's very close to the zinc binding site, and the fluorescence intensity of that tryptophan changes dramatically when zinc binds. So we can mix them together, holo AZTT, APO AZTC, and put them together, and you can even monitor the kinetics of transfer. It's pretty slow, actually. It's like seconds to maybe a minute. But if you do that, if you put them together and then separate, and then like you can separate them very easily chromatographically, which is one of the other ways that we showed transfer, is that, you know, you can put them in the same tube. The zinc is on the AZTD. There's no zinc on AZTC. You put them together, even in the presence of high concentrations of EDTA, and you get transfer. But then you put them on the column, they separate, right? So I believe it's probably transient. AZTD is a little mysterious functionally too. You can actually cut it out of the genome, and they don't seem to care that much. Whereas if you remove the solute binding proteins, they're very sick. So I'm not sure exactly what it's doing in there, but it does transfer, at least in vitro for sure.
Very interesting. All right, one final question from Chu Young Kim. Eric, can you show us the slide that has a picture of the crystal obtained with the tungsten compound? They have a question about the UV image, slide 16.
Okay.
Is that a salt crystal at 12 o'clock?
You know, I don't see it in the UV, so it could be. It also, you know, these images can be a little tricky to look. It might be on edge, and then we're just not seeing it. But yeah, it's definitely possible because it doesn't light up. And if I should have put the second harmonic image in here. I don't remember if it shows up there either. Also, hi Chu. Chu's in El Paso, so he's like my neighbor, as close as neighbors can be out here.
Yeah, it looks like it's almost out of the imaging plane of the other crystals, so it could just be that it's not quite on the same focus there.
Right.
All right. And then another one from John. I think this is our probably last one due to time, Sarah, right? Okay. Hi, Eric. Nice talk. You mentioned you had some crystals that you were not able to solve using molecular replacement. Have you had any luck using AlphaFold 2 models?
I have not tried that. That was before they were so readily available. So it seems like just within the last 6 months, if you go into Unipro, every one has an AlphaFold model.
Yeah.
I actually talked to somebody at the ASB&B meeting, and that's pretty much true. They basically added them to everything, which is super cool. I have heard of that working for sure. I think you'd have to be a little careful. An AlphaFold is nice in that it gives you a degree of confidence, right? It's like color coded. Where you have flexible regions, let's just, for example, say, yeah, things like this is where it'll fail for sure.
Yep.
Because, and then even that, you know, how physiologically relevant is that orientation of the loop? Can't really say. It's probably crystallization that makes it stick in that conformation. It is a conformation it can adopt, right? But is it the way it is most of the time? I don't know. But I have heard, I've seen papers where people do it. So it's definitely been done. Just not by me.
Awesome. Well, let's all thank Eric once again for his talk. We appreciate you being here and for all your shout outs to the Crystallization Center.
Oh, sure. Yeah, it's been great for us. So hopefully this membrane thing is going to take off, because that would be awesome.
Totally.
All right. Thanks, everybody.
Thank you, Eric. That was really great. And so next up, we have Crystal McLaughlin from Vassar College. She's going to be talking about some of her research. And so, Crystal, if you want to go ahead and share your slides. And it
Hi,
looks like,
everyone.
there you
Just
go.
gonna start the slideshow. Hi.
Perfect. Okay.
Okay.
Thank you.
Okay. Yes. Thank you so much for inviting me here. I really enjoyed the last talk, Eric. So I am going to talk about some of the things we started here in my group, and we haven't gotten to the point yet of crystals, like at all of the success, but I really hope we're on the path to there using the National Crystallization Center. So I am Crystal McLaughlin. I'm in the chemistry department at Vassar College. We are a small liberal arts college. We don't have any graduate students or postdocs. It's just all undergraduates and myself. So all of the work I'm going to talk about today is being done by undergraduates. And in that poll that Sarah asked about how do you set up crystal trees, we also don't have any robots or anything like that. We have undergraduate hands setting up our crystal trees. So we are very excited to take advantage of the services from HWI, so the National Crystallization Center, because it really allows us to expand what we've been doing. So let me go ahead and tell you a little bit about it and how that has come into like sort of helping us so far in the progress that has been made. All right. So my title is Structural Insights into Conjugative Antibiotic Resistance Transfer. And so my lab studies a lot of microbial proteins. And one of our projects is looking at how bacteria get antibiotic resistant genes through horizontal gene transfer, through conjugation in particular. So why do we care about this? So more antibiotic resistant strains emerge each year. And so it's an increasing problem. One example that is pertinent to college students that I like to talk about when we're sort of bringing students into the lab, and now I talk about it in my talks, is drug-resistant STDs. In particular, drug-resistant gonorrhea is on the rise. And so this is a graph from the latest CDC antibiotic resistance threat reports, just showing that there are many, many emerging strains of gonorrhea that have more and more drug resistance. So you can see all the drugs below in the different colors. And what is also really alarming sort of about this this chart is that we can see that since the 1980s, we've actually had to sort of retire 4 different antibiotics that used to be able to treat gonorrhea infections. And so we're at 35% now that are sort of like all drug resistant, and it's just going to keep going up. So the question we want to address in our lab, like have our research contribute to, is there some way that we can reduce the spread of antibiotic resistance? So is that possible? Can we at least be able to stop these resistance from spreading through the community so that our existing antibiotics can continue to work, right? So we're not creating as many new antibiotics. Bacteria can get these resistances pretty easily. So trying to reduce the spread is one thing in our toolbox for fighting bacterial infections. So how do we do this? How could we even reduce the spread of antibiotic resistance? Well, first we need to learn more about how bacteria do this. How do they actually spread it around? And so one of the major ways that bacteria can get antibiotic resistant genes is through extra chromosomal plasmids. So I'm showing here a really like basic mock-up of a bacteria. It's in blue, and then you have its chromosomal DNA, and then it usually has like extra chromosomal pieces of DNA, often in the form of a DNA plasmid, shown here as a double-stranded like black circles. And so the transfer of these plasmids is what we study in my lab. So conjugated plasmid transfer is the name for the process whereby these plasmids can be shared between 2 bacterial cells. So I'm going to give you an overview of the process, and then we'll go in a little bit more detail as to the proteins that are doing this job. So in this schematic, we have now 2 bacterial cells. One of them is we're going to call our donor because it has a plasmid that has antibiotic resistance on it. And the other one we're going to call the recipient. It's going to receive a copy of this plasmid. So this is very much glossing over all of the details. Conjugation can happen if they come together. And then what you kind of have is now there's a channel or like a pore between 2 bacterial cells that I'm just illustrating here as this sort of gray ring. And so this I've labeled T4SS and T4CP. So there is a secretion system. That's what the SS stands for. And in this case, the ones that we study are type 4 secretion system. And then there's a coupling protein, a type 4 coupling protein, that's the CP, that couples the activities of the secretion system to another really important complex called the relaxosome. And the relaxosome is actually what we're trying to figure out in my lab. So it's this multi-protein complex that processes the plasmid. And so the relaxosome will bind to the plasmid at a place called the origin of transfer. It will nick the plasmid, it will unwind one strand, and then it will bring that strand to the secretion system and coupling protein and allow it to be fed through to the other cell. And so just one strand goes into the other cell during conjugation. And then what happens is that once that single strand of DNA is detected in each of the cells, because now you've left behind one unnicked strand in the donor, they get recircularized by the cell's machinery. And then at the end, you have a donor and another donor. So now you have two antibiotic resistant genes. And so we're really interested in that process and the proteins that do the processing of the plasmid. So if we zoom in a little bit more to look at the relaxosome, like I mentioned, is this multi-protein complex, and it's usually composed of a relaxase, a helicase, and an accessory protein or multiple accessory proteins. In the system that we are studying, and I'll talk more about that a little later, we have kind of an idea what's going on, which is that here in this schematic, I'm showing the type 4 coupling protein and the type 4 secretion system here, this light pink and this gray. And then the plasmid DNA is shown here in orange. And I pointed out here the ORET, which is the origin of transfer. It's a consensus sequence where the nick happens. And what we think happens first is that a host encoded protein called integrative host factor binds the DNA and introduces a bend, which is probably helping the nick happen. Once this happens, TRA-K, which is an accessory protein shown here in dark pink, will bind to two places on the origin of transfer, and recruit TRA-I, which is a relaxase and helicase protein, a bifunctional protein fused together. And these components together, we think, form the relaxosome. And then once that happens, unwinding starts, and then we know that TRA-K and TRA-I, so the relaxase, the helicase, and the accessory protein all interact directly with the coupling protein, and some of them with the type 4 secretion system. So this is what I am trying to explore structurally, and there is no structure of a relaxosome at all. So there's none intact. There are a couple individual proteins that have been solved. There's two TRA-Is, one full bifunctional protein. Really, the structural information for this complex is really lacking. So this is where we are trying to contribute through our research. So on our plasmid, the really cool thing about conjugative plasmids and why they're conjugative is that they encode the genes for their transfer. So the transfer genes that I mentioned are encoded on the plasmid itself that's going to be transferred by those proteins, by the relaxosome. So I'm showing here just a kind of mock-up of the important parts of the plasmid. Of course, you have your antibiotic resistant genes. Then you have the origin of transfer, which is that consensus sequence where the nick happens. And then right next to the origin of transfer, you usually have a bunch of genes that are referred to as the TRA genes, so in the transfer region. So we are studying two model plasmids in my lab. The first one is from Salmonella typhimurium, which the explanation of TRA-K and TRA-I and what I talked about before, that's what we think happens in Salmonella. And then we're also studying a plasmid from Staph aureus. And I will say that we know much less about what is happening in Staph aureus in terms of this relaxosome complex forming. So first, let me tell you a little bit about our work in Salmonella. So conjugated plasmid transfer in Salmonella, like I said, we know a little bit more about it. It's been not pretty well studied, but somewhat studied. And Salmonella typhimurium, which is where this plasmid is isolated from, is the causative agent for a lot of foodborne illnesses, some sort of gastroenteritis and things like that, which is why you shouldn't be eating raw chicken and things like that. Our plasmid is called PCU1. It was isolated all the way back in the 80s. That's a pretty broad host range, which makes it interesting to study because it doesn't just exist in Salmonella. It can actually exist in other related strains like E. coli, and it confers resistance to 3 different antibiotics. So one of the reasons I chose to study PCU1 is because it has a pretty small transfer region. as I explained in the assembly of the relaxosome, we think only TRA-I and TRA-K are the two transfer proteins that are needed. In some other plasmids, this transfer region is different and encodes many more proteins than just this two. So this is sort of our first simple model to look at. So TRA-I, as I mentioned, is both a helicase and a relaxase, so it nicks and unwinds the plasmid. And then TRA-K is one of the accessory proteins. So today I'm just going to talk to you a little bit about our work trying to structurally characterize TRA-K. So what we did first, once we were able to purify TRA-K, was we did some AFM experiments, so atomic force microscopy, which sort of gave us the overall shape and told us where it was binding on the plasmid. So here is an image from some of our AFN experiments. And what I'm showing here, all these bright dots are basically the protein on a mica surface. And so this is TRA-K alone, so it's not bound to anything. If we zoom in on one and we were able to do volume analysis on the individual particles, we realized that it was forming a stable tetramer, which is kind of what we saw from the purification process, that it was four of the protein together, and it's pretty well ordered. All of them are tetramers, so it seems to be a tetramer, which was really cool to see. And then we took the part of the plasmid, the origin of transfer, so it's 381 base pairs, and we wanted to see where might TRA-K be binding. And in fact, if you look at this image, and I circled a couple of them, we saw that TRA-K was binding to 2 different places. So that sort of pointed us in the right direction as to which pieces of, or which sequences on the origin of transfer is bound to. And this was actually kind of hard to do because, like I said, there's not a lot known about these accessory proteins or the relaxosome in general. So we didn't know exactly where it would bind. So this allowed us to be like, okay, there's 2 sites. There's another accessory protein that does something similar to this. So we were then able to try to pinpoint where it bound. So TRA-K lacks pretty much sequence identity to any other accessory protein that we know, so any other kind of relaxosome protein. But based on the AFM, we were like, maybe it has a similar structure to one of the few solved accessory proteins. So in this case, I'm showing the structure of F plasmid TRA-M, which was solved several years ago now. And F plasmid TRA-M has these 2 like sort of bundles down here, DNA binding, helix turn helix. Actually, this one is not a helix turn helix. Ours is predicted to be a helix turn helix. So this has some other kind of binding mode here. And when we look at the AFM image of PCU1 TRA-K, it kind of looks like a little boot to me, which is actually a little bit what I feel like TRA-M looks like. So I tried to draw a little boot here, which is if, you know, we can see that there's a lot of flexibility here in the region, the loop sort of connecting the DNA binding domain to the tetramerization domain up here. So TRA-M is also a tetramer, which again was very like validating for us seeing this data. And so we think maybe TRA-K will look something like this. We don't have the structure yet. This is as good as we can get right now in terms of resolution. It sort of looks like this. So we thought then what might be good is to use the DNA to stabilize TRA-K, because it looks like those DNA binding domains, if it looks like TRA-M, will be very flexible. So if it's bound to DNA, it'll trap it, and hopefully we'll get a crystal. So we found one site that bounded really high affinity that we started using in crystallographic studies. As I said, the PCU1 origin of transfer is about 381 base pairs wide. And we tried several different sites based on what we saw in AFM. So site A, site B, and site C. And then this color coding shows you where the other components of the relaxosome will bind. So like where IHF binds is shown in a dark black. And then the origin of transfer, or RET here, this little dark purple, this is where the nick happened, the nick site, and that's where TRA-I would bind. So we're trying to figure out where would TRA-K be. And of course, I guess the natural answer would be to do like a DNase 1 footprinting assay, which we eventually did, but it was after this sort of looking at it, and it confirmed actually what we saw based on the AFM as well too. And so site B, it bound with pretty high affinity, so the KD was 726. It also bound to site C at a lower KD, but we decided to move forward with crystallographic studies using a 25 base pair sequence of site B. So that's this pink site here. So I came to Vassar like about 5 years ago, and we started doing these optimizations from screen. So my first student worked on this. So this is Isabel Saccharin. She graduated in 2019. And so she sets up a 96 well tray, and she uses the Jenna Bioscience nucleic protein nucleic acid screen. And we actually get like this hit, which at first I was like, that's very tiny. I don't know if that's real, but she was able to optimize it to sort of look like a real crystal, which was very, very exciting for us. And this was the first thing we tried to crystallize. And so again, I did not believe that it was happening. But here I am 5 years later. So I feel like maybe that that worry was a little warranted. But it did turn out to be a protein crystal. However, these actually didn't seem to be in the metastable zone. They dissolved a few days after. So even though we were able to grow them, they weren't very stable. So after Isabel graduated, the plan was to bring more students onto this project, but you may have heard that there was a pandemic. So we actually had to shut down this project for about 1.5 years. And then we restarted it in 2021. So then 2 new students came onto it, Lilith and Jordan. Jordan is still here working on it. And they worked really hard to change the purification of the protein itself. And then they also did, different drop ratios, things like that, trying to figure out if they could grow better crystals using this. And so in fact, we used a different screen as well, and we were able to grow what seemed to be like better crystals. They were definitely more stable. They were long lasting. So they grew some that looked like these, a lot of these diamond shaped ones. And then actually one that looked like this, which I just thought was hexane diol crystallizing out of solution, but it turns out it wasn't. But so they got a couple different crystal forms. So we were very excited when we sent this off for beam time, and nothing diffracted, none of these very beautiful ones, which we even grew much bigger. I just don't have pictures of that, but there's no diffraction. And then very late last year, we got diffraction from one of these, but it was not enough to like do anything, no indexing, nothing, but it seemed to diffract. And so we're able to grow all these crystals and nothing's diffracting. So then I said to the students, you know what, let's try the National Crystallization Center to see if we can find a couple of different conditions and see if we can maybe get better crystals that way. So we sent off trachea and DNA to HWI in like last October. So late in the year, I think it was right before winter break, which because we're an undergraduate institution, then winter break happens, all the students go away and they don't come back to like January this year. So really we started interpreting that data this semester. And so we got a couple of different hits. And I'm not showing the UV images, but all the hits that I show here today have basically positive signals in both the UV and SHG. So here's one. It kind of looks like our diamond crystals again. So I'm still a little skeptical about will it diffract or not. These are starting to look a little different, but still very same. But the condition is very different from the one that we had, although everything seems to be crystallizing in PEG. So these are the 2 that we're following up on now. And I know we had a little bit of discussion about microbatch under oil, and most people use, sitting or hanging drops of vapor diffusion. We also use vapor diffusion, and normally we do hanging drops. So actually we tried that first and we weren't able to regrow any of these. But just last month, we learned how to do microbatch under oil. I had never done it before, so I was a little bit hesitant, but my student figured it out. I basically told her to go read the HWI's guidelines, see if we can do this. And she was able to regrow one of these. So not the sodium citrate condition. So we just got these. They were grown about a month ago. And we realized though they are also not stable because they've already dissolved as of today. So she's still in optimization, and we're hoping that we'll get some crystals to send to our next beam time, which is actually in May. So we send all of our crystals to Lawrence Berkeley National Lab out there. So that's our one project that hopefully will become very successful for us once we get those crystals out. And then I'll tell you a little bit about what we're doing on the second project, which is a little bit like less known as well too. So the second project that we work on, like I said, is a plasmid from Staph aureus. The plasmid is called PSK41. So Staph aureus is in the microbiome of pretty much most healthy individuals. It's like in your nose and your skin. It's really an opportunistic pathogen. So if you get a cut or something like that, it may give you an infection, like a staph infection. And so MRSA is sort of the most famous of the staph infection because it's a serious threat as classed by the CDC. The PSK41 is an interesting plasmid to study because it's the largest plasmid family in all of Staphylococcus. And so it is the founding sort of member of the plasmid that carries methicillin resistance. And in fact, other related plasmids in this family can also carry vancomycin resistant. So then you can get something called VRSA, which is vancomycin and methicillin resistant staph, which is not great. So understanding how PSK41 is moved around staph like cells would be really useful as well. The cool thing about PSK41 is that it's this really small plasmid comparative to other plasmid. It only encodes 42 genes. And remember, they encode all of the proteins for their transfer in that transfer region. So we were hopeful that we could figure out what the relaxosome looked like in staph fairly quickly. But that is turning out to be a more difficult problem than we anticipated. And this is because most of the proteins in the transfer region, so right next to its origin of transfer, don't match any of the other proteins we know. Only the relaxase has been confirmed and discovered. And so there's a structure of relaxase, but this relaxase in staph does not have a helicase attached to it. So we're still looking for the helicase and still trying to figure out which proteins are in fact accessory proteins. So there's a lot less known about this conjugation in staph. So we're still trying to identify and verify protein functions. We don't do a lot of this verification of the function is done by our collaborator Neville Firth in Australia. So he's at the University of Sydney. So he works on staph, and we sort of take what are interesting targets from him and try to crystallize them. So we still don't know what are in the relaxosomes. We had one guest that we're working on right now, which is ORF90. And so the reason why we chose ORF90 is based on it was conserved in all of Staphylococcus. So all of the plasmids have this protein, so it must be important in some way. It's right next to the relaxase on the plasmid, as well as in the origin of transfer region. So perhaps it plays a role in the relaxosome. However, as I mentioned, there's no sequence similarity to known proteins. So this one is even more of a mystery than TRK. TRK at least has a region predicted to be a helix-turn helix, so we think it binds DNA. This one has no predicted conserved motifs, so we don't know what it looks like. So I have two students that were working on this, Vanessa Gonzalez and Kate Enquist. And so they have been trying to figure out how we can solve the structure of this to give us some more information about what it does. Now, I had started this project several years ago before the students came on, and this was one of the hits that I got from an NCSG4 screen. And so these crystals, really cool looking, they looked very favorable, like they might diffract. And they in fact did diffract, but they diffracted to 4 angstrom, so 4.09 I think is the most I could get. So here's some of my data from trying to refine this 4 angstrom data set. However, this protein seems to just be made of helices that bind together. So I cannot figure out where it starts or ends. And since the resolution is so low, I can't build any side chains. So I spent a lot of time trying to figure out this data. I don't know where the N terminal is from the C terminal or anything like that. Seems to be some kind of dimer, and we can see that from gel filtration studies as well too. But this polyalanine model that I built is sort of as far as we could come, and we wanted to figure out, can we do something else to try to solve this structure. So it looks like it might be interesting in some way because I just could not figure out what that was. So we created a new construct that the students started working on last summer. And so they were able to clone and purify a ORF90 construct that was tagged to MBP, which we are not going to cut off. We're going to leave the maltose binding protein MBP on ORF90 to see if that helps with crystallization. And then we use that for trials by sending it out to the National Crystallization Center. And this is because my students tried in-house and they got not a single hit, not a single thing grew in any of their wells. So we sent it out and actually started getting, we have a bunch of hits that we want to follow up on. So here's one. So it's got a positive signal in all of the UV as well as the bright field. So this is sodium bromide, CAPS, PEG 1000. Here's another ammonium chloride and PEG 1000 and tris. There's another kind of needle looking one. And the one that my students are working on now, which sort of look like the biggest crystals here, are lithium bromide, tris and PEG 1000. But there are a lot of these. So actually we got a lot of things that look like they're crystalline and they're protein. And so that was really exciting because we had 0 hits from in-house. So this is where we're at with this project though. The students are abroad this semester. So this summer, we're going to work on trying to regrow these hits. Now that we know how to do microbatch under oil, we will be doing that as well too. And then hopefully sending some crystals out and getting some structures soon. But it's been really great, having the National Crystallization Center as a resource, especially at a really small school like ours where we only have undergraduates. It's really hard to set up a lot of these trays. So let's purify a lot of protein. So the ability to set up the smaller drops is really, really helpful because we just don't have that much protein. Okay, so in summary, we have these new crystallization conditions for these 2 proteins that we're working on from different plasmids. So we're very excited that that will pan out into hopefully structures that will give us some more information. And so the next step, of course, is we're optimizing and we're going to send for data collection. And then further down the road, what we want to do is just purify the rest of the relaxosome components, which I've listed here, do some pull down binding experiments, and really then try to assemble the relaxosome so we can get a view of the full structure. So we have a lot of cool experiments planned. Thank you all for your attention. Thank you to the organizers for inviting me. Thank you to my students for doing all of this work. I did not set up any of these crystal trays. They did it, and they're really awesome. Yeah, so if you have any questions, please let me know.
Wonderful. Thank you so much, Crystal. It sounds like you have a lot of great work going on in your lab and a lot of dedicated students. So that's wonderful to hear about. I have a question to kind of start off with. And my question is about the first project that you discussed. I'm curious whether you've tried to kind of fiddle with the DNA length in your crystallization sample to try and influence the formation of the crystal lattice.
Yes, that's something we want to do. The only thing we have done, and so I didn't mention, but so these hexagonal crystals are actually a different piece of DNA. It's the site C, the second site. So I just started doing that. But yeah, I am aware we haven't tried overhangs or anything like that yet, but that is the next thing if this does not pan out. And really though, I hope to just get the complex. So I'm really interested in trying to see how the proteins interact with each other and the DNA. So I think if the next big step for us really is trying to put them all together, because that's really what we want to see as well too.
Awesome, very exciting. All right, a question from Christina Bourne in the chat. They ask, that is really cool to use AFM to see these, I agree. What would be a limit for resolution by AFM? And is it dependent on the AFM tip or something else?
That's a great question that I don't know the answer to, but because I did this in collaboration with the Erie Lab, and now I'm working on AFM experiments with my colleague down the hall, Zach Donhauser. So I don't know what the resolution limit is, but I can send you a really cool cool paper that I found last year that I gave to Zach and we're very excited about, where you can actually get very close to atomic resolution from AFM by like how you process your data. So I'll send it to you. So I don't know the answer offhand, but it's getting better and better. So we were going to try to do that once we collected more data from this. Yeah.
Awesome. Miranda asks, have you explored other structure methods such as small angle X-ray scattering to help with some of these intractable structures?
No, I haven't. I don't know where I would go. So I suppose one of the things that being at a small college and we're kind of far away from a lot of big universities, I guess New York City would be the closest, but it's still kind of far, is that we don't have access to a lot of these great, I guess, pieces of equipment. And since I'm not an expert in that, I haven't really done very much small angle scattering, and nobody here does that also. I haven't branched out into that yet, but that is definitely something that probably would be really good for the complex as well too, which we're still trying to figure out. Is it dynamic or is it stable, right? Do all the proteins stay together or do they come and go as the process goes on? So I suppose if I had a collaborator, yes, but right now there's just nothing close to us that we can go to do that. And I don't have that expertise yet.
All right, next question, Manoj in the chat. I think this is in reference to the ORF90 project. They ask, have you tried adding DNA?
So that is a great question. And so I did, in a moment of desperation, throw ORF90 in with the TRA-K DNA, and it bounded. So it does have some sort of DNA binding domain in it. But and then I set up crystal trays with that, and I did get a hit. So I didn't follow up on it though, because as I thought about it, I'm like, what would I say in this paper that this is a random piece of another plasmid that I threw in? So the next step here, what we want to do is send it out so we want to do DNase 1 footprinting. I want to do that so we get the real piece so I can put it in so that I can really talk about this is where it binds. But that's a great thing that we thought about, but I just don't know where it binds on PSK41 yet. And I kind of put the pause on our crystallization with the random piece of DNA. Although we know that it does bind, but I have no idea where it binds. I'm like, what is this? I don't even know. Yeah. So great suggestion.
Yeah. It's great to hear too that you're not subjecting your undergrads to tons and tons of EMSAs. That's what I had to do when I was an undergrad.
Right. Yeah. No. Yes. So I didn't mention, but we won a Cottrell grant. I won a Cottrell grant last year, and so now I've been outsourcing a bunch of stuff to try and give us a little bit more, I guess, of a push, because we just don't have access to the kinds of equipment you would at bigger institutions where you would have labs that also maybe had like gels that you could do the DNase 1 footprinting, or we don't even have like a radioactivity place where I could run that kind of gel if I wanted to. So we found a company that does that. So we did it for TRA-K. And like I said, it was very validating to see that what I came up with was actually where it bound. So the results just all match. But we are in right now, we're just having an issue purifying with the protein, as sometimes that happens. And so we're trying to get enough to send out for that, while also saving it to set up more crystal trays. So I'm trying to figure out that balance there. Yeah. But yeah, not too many EMSAs here.
Ankit asks, have you tried using antibody fragments as crystallization chaperones?
I have not. Hi, Ankit. That is not something I have done before, but I've seen a lot. I see that done a lot, but yeah, I haven't even tried that or even thought about doing that. But I suppose as we get deeper, especially into the complex, that would be really great as well too, if it needs something to stabilize it. Oh, great suggestion.
Well, it sounds to me like you're using MBP kind of as a crystallization chaperone in your ORF90 project as well. So
yeah.
All right, and sorry, go ahead.
No, that's what we're trying to do with that. Yeah, the other thing is I haven't tried to bind I don't really know much about like trying to bind ORF92 DNA with MBP bound to it. That we haven't tried yet as well too. So other things, yeah.
And then a last question from Anwar. They ask, will AFM work for large, complex, multi-domain proteins?
Yeah, I do believe so. Yeah, so the Erie lab does a lot of the DNA repair systems, and I know that those proteins are pretty large and have multi-domain. And so they are the ones we first started working with that protein on. So I think it does work. And if you want, I can send you that paper that I'm going to send to Christina as well too, about like the advances in processing AFM for protein structure and trying to get some of those more atomic level details.
Wonderful. Well, thank you so much. I really enjoyed your talk.
Thank you so much. All right.
Yeah, let's everybody thank Crystal again. Thanks so much. That was really awesome. So we are now at lunch, our lunch break. We do have, I'm going to share a poll, and it's just a poll for find out where attendees are actually from. So if you click on the link, it should take you to the poll, and then I can share the screen. But we will reconvene at 12:30. I'm also going to put a link in to the chat for our Crystallization Center survey. And so we'll be sharing this again later, as well as via email. But we do encourage people to fill out the survey, and I'm going to actually put it in correctly. Okay, there we go. And so we look forward to reconvening in about a half an hour. And in the meantime, again, I will share that survey if people are entering it. So. Okay. Okay, and I'm just trying to share. Sorry, there we go. And I'll do this again at the end of. It looks like people are from all over. There's someone from Potato. So with that, hope everybody has a good lunch. I will kind of put this back up after we're back from or well, lunch, dinner, breakfast, wherever you're at. Have a cup of coffee. Okay.
Enter your PIN. You have 2 new voice messages. To play new messages, press 1. New messages. Message 1 from 845. 5463742 received.
Hi there. Does somebody need to mute